Can plasma therapy cure COVID-19? Let's see. What is a plasma? Whole blood when we centrifugation. Its separate, upper part contains plasma 55% of total blood. Middle part contains buffy coat leukocytes and platelets less than 1% of total blood. Lower part contains erythrocytes 45% of total blood.
Plasma is the protein-rich, gold-colored liquid portion of blood that remains after red blood cells, white blood cells, platelets, and other cellular components. Plasma, which is used to make life-saving plasma protein therapies, is collected through a process known as plasmapheresis. During this process, an automated device is used to separate the plasma from the blood. Once the plasma is collected, red blood cells and other components are returned.
How to test COVID-19?
COVID-19 is an infectious disease caused by severe acute respiratory syndrome coronavirus 2. When a person is infected, the most common symptoms include fever, cough, and shortness of breath. To start a test, the samples can be collected by a nasopharyngeal swab or an oropharyngeal swab. For nasopharyngeal specimen, the swab is inserted in the nostril and gently moved forward into the nasopharynx, then it is rotated for a specified period time to collect secretions that contain the virus. Once the swabbing is applied, the swab is placed immediately into sterile tube containing a viral transport medium. The standard method of coronavirus testing is polymerase chain reaction, PCR, which is a method that used widely in molecular biology to make millions to billions of copies of a specific DNA fragment rapidly. Coronaviruses contain an extraordinarily long single-stranded RNA genome. To detect these viruses with PCR, RNA molecules must be converted into their complementary DNA sequences by reverse transcriptase. Then the newly synthesized DNA can be amplified by standard PCR procedures. This approach is universally known as RT-PCR. To perform this method, basically viral RNA should be extracted. Several RNA purification kits are available for convenient, fast and effective isolation. Preparation of the reaction mixture for PCR amplification. In this step, a master mix is used, which is a pre-mixed concentrated solution that consists of buffer, reverse transcriptase enzyme, nucleotides, forward primer, reverse primer, Tachman probe, and DNA polymerase. Finally, to complete this reaction mixture, the RNA template is added. The tube is mixed by pulse vortexing. Then the reaction mixture is loaded into a PCR plate, which generally contains 96 wells, allowing the analysis of several samples at the same time. Next, the plate is placed in a PCR machine, which is essentially a thermal cycler. For the measurement of the fluorescence signal, a tungsten halogen lamp, an excitation filter, mirrors, lens, an emission filter, and a charge-coupled device CCD camera are used. Filtered light from the lamp is reflected off mirror, passes through a condensing lens, and is focused into the center of each well. Then fluorescent light emitted from the wells reflects off the mirror, passes through an emission filter, and is detected by the CCD camera. In each PCR cycle, light from excited fluoriphore can be detected by the CCD camera, which converts the light that it captures into digital data. This method is known as real-time PCR, which allows the monitoring of the progress of the PCR reaction as it occurs in.
The therapy entails giving patients a transfusion with plasma or serum from those who have developed antibodies to a virus or bacteria. This process grants the patient some passive immunity. Convalescent blood is an option if there are no medicines or vaccines to treat an infectious disease. The first valid trial was done in 1892 for diphtheria, using serum from animals.
COVID-19 pandemic. With the Indian Council of Medical Research approving the clinical trials, doctors are now dusting off a century-old treatment to battle COVID-19. Convalescent plasma therapy. So what is CPT and how does it work? CPT uses antibodies found in the blood of people who have recovered from an infection to treat patients who are newly sick. Developed in 1900 by Nobel laureate Emil von Behring, This method was earlier used during the 1918 Spanish flu pandemic. It was also used during the Ebola pandemic, which started in 2013, and in 2003 against SARS. We know plasma is one of the 4 components in our blood, and along with carrying cells and protein throughout the body, it also produces antibodies to fight pathogens. Well, in this case, the coronavirus. In CPT, blood is drawn from a person who has recovered from the disease. Then the plasma is extracted from the blood and is transferred to treat other patients. Doctors think that after someone infected with a virus like the coronavirus recovers, their blood is rich with antibodies that their immune system produced to help fight the virus. States like Kerala, Gujarat, and Punjab have already started to use plasma therapy for COVID-19 patients. On April 21st, a 49-year-old man who had tested positive for COVID-19 and was on ventilator support at the Max Hospital, New Delhi, became the first patient in India to be administered plasma therapy. According to several reports, he has been showing progressive improvement and is faring well. While it remains to be seen if this age-old treatment will be an effective treatment for the deadly virus, medical experts are of the opinion that it can be a bridge until a vaccine.
Side effects. No definitive studies exist showing effectiveness. In case of dengue, convalescent serum was found to make patients worse, as it led the virus to replicate. There could be transfusion-associated reactions. Unknown pathogens could be transferred into a patient during transfusion. To be continued. Do check out my video and subscribe to my channel. I would like to hear your feedback. Give support to me by subscribe my YouTube channel.